Hello, how are you? So today's lesson we are going to learn about the structure of a bacteriophage, and before we do that we have to know what is this particular bacteriophage. So basically a bacteriophage is a phage, is a type of a virus that infects and replicates within a bacteria. So the two words bacteriophage is derived from two Greek words, bacterion meaning bacterium and the phage meaning to eat, so that when the two words are combined it forms a bacteriophage. So we are going to look at the structure of this bacteriophage, and as you can see from this my diagram, we have the structure. So we do have the head, the capsid. The head is also known as the capsid. So this is the capsid, the upper part. So the capsid here, the whole of this region is called a capsid. So the capsid here contains the nucleic acid. As you can see, the upper part is the nucleic acid. So the main function of this capsid or the head, it is always made up of protein and it always encloses the phage's genetic material. So any bacteriophage has a genetic material, and the genetic material is always either DNA or RNA. The capsid encloses those genetic materials within them. Nucleic acid, that is whereby the genetic materials are composed. And I said earlier that the genetic material is either DNA or RNA. So the main function of this genetic material is to encode the instructions for viral replication and assembly. Encoding instructions for viral replication and assembly. Then we also have what is called the tail. The tail, as you can see from the diagram, the tubular part which is blue, which runs from the upper part until the lower part. So it runs from the bottom of the capsid, from the bottom part of the capsid up to the lower part. So this part here is called the tail, and majorly the main function of the tail is to deliver the phage's genetic material into the host cell. So this is the tail that is going to deliver the genetic material into the host cell. That is the main part, the main function of the tail. Then from there we do have what is called the base plate. So the base plate is just the lower part of the tail. We have what is called the base plate. As you can see, it is in this lower part. So this is called the base plate. And the main function of the base plate, it always serves as the interface between the tail and the host cell. So this base plate serves as an interface between the tail. The tail, I said, it is this tubular part running from the base of the capsid to the lower part. So the base plate main function is that it serves as the interface between the tail and the host cell. From the base plate we do have what is called, again, the base plate. Another function of the base plate which you can say is that it contains various proteins which is involved in host recognition and also DNA injection. Contains various proteins involved in host recognition and also DNA injection. Then from there we have what is called the tail sheath. The tail sheath, This is where the tail sheath is located. The tail sheath. So the tail sheath is just located, it is attached to the tail. It always surrounds the tail tube. So the main function of this sheath is to contract during the process of infection, and during that contraction process, it will allow the tail to penetrate the bacterial cell wall. So during the contraction, it will allow the tail to penetrate the bacterial cell wall. So that's the main function of the sheath. Then we also have another structure of the phage, bacteriophage. Basically, it is not a structure, but you realize that some bacteriophage do have the enzymes. For example, the lysoenzymes. And these enzymes, their main purpose is to help degrade the bacterial cell wall. They degrade the bacterial cell wall, facilitating entry into the host cell. So that is the main function of the enzymes which are found within the bacteriophage. So that's what I basically want to learn today. And from this my slide, I just said that we have the first part is the head, and I said that the main function of the head is to enclose the phage's genetic material. And I said that this head contains a protein code. So the head or the capsid is always made up of a protein. And the main function is to enclose the phage's genetic material. Again, I said earlier on that the genetic material, which is also found inside the head. So the genetic material, I said that it is either DNA or RNA. And these genetic materials contains the phage genes, which encode the instructions for viral replication and assembly. So the main function of those genetic material is to encode the instructions for viral replication and assembly. Another structure which we have observed is the tail, and I said that the tail is the tubular structure. The tail is the tubular structure. That extends from the head of the phage. Again, I said that the main function of that tail is to attach to the bacterial host and delivers the phage's genetic material into the host cell. So the tail is the one which is going to attach to the bacterial hosts, after which it will deliver the phage's genetic material into the host cell. And I said that the phage's genetic material is made up of two, the DNA or the RNA. So from the tail also we discussed the tail fibers, and I said that the tail fibers are always spikes. They are like spikes, and they're always attached to the tails. So if you can see from the diagram which we observed, the tail fibers, you do have these long tail fibers. So the tail fibers, they're like spikes, and they come from the tail itself. As you can see, they come from the tail, and their main function is to recognize and attach to specific receptors on the surface of the bacterial host cell. Again, we have discussed about the base plate, and I said that the base plate is always located at the end of the tail. The base plate is located at the end of the tail. and always it contains various proteins which is always involved in host recognition and DNA injection. Then we do also have the tail sheath, which you have observed earlier on, and I said that the main function of the tail sheath is to contract during the process of infection, and during that process of contraction it will allow the tail to penetrate the bacterial cell wall. Then I said that some bacteriophages can carry enzymes like the lysozymes, and these enzymes mostly helps to degrade the bacterial cell wall, facilitating entry into the host cell. So that's one are the major structures of the bacteriophage. And in our next class, we are going to discuss the life cycle of this particular bacteriophage. That's the end of the lesson, and thank you.